The techniques of genetic modification of DNA have the potential for chaos if they are not approached carefully. The idea of changing the genetic structure of an organism, seen by many to be a violation of the natural laws of the universe, is not a particularly new one. It began with test tube babies in the 1960s, and has come as far as the crossing of genetic blueprints of different species. In fact, the creation of these chimeras is already underway. The legendary Greek chimera was a terrifying beast with the body of a lion, the wings of an eagle, the tail of a serpent, and the head of a dragon. This is exactly what scientists seek to do with the current tools of genetic modification, the bridging of interspecies boundaries. In effect, scientists wish to create their very own chimera of legend, hence the name of these new cross-species organisms. On January 11, 2001, quote, researchers in Oregon produced the world's first genetically altered primate by inserting a jellyfish gene into the embryo of a rhesus monkey, a development that brings gene slicing experiments closer to humans. End quote. Sheep and goats are also being spliced with human DNA for transplant purposes. This field of genetic research, especially involving the modification of DNA, is not regulated to maintain the best interests of the people. In fact, it's constantly being interpreted as an attempt at eugenics, which is the science that deals with the improvement of races and breeds, especially the human race, through the control of hereditary factors. Scientists are perceived as trying to create the perfect being, a human being with no weaknesses and perfect for evolutionary advancement. Scientists are accomplishing this goal through the selection of precise genes intended to equip this new organism with the desired traits. Many are still asking, how is this done? How are scientists fiddling with DNA? Well, the answer is smaller than you think. In fact, it's microscopic. As previously mentioned, the most common technique for the splicing of genetic material is through viruses and bacteria. The structure of a virus is perfect for the assimilation of DNA segments. This is actually how viruses infect their hosts, by injecting the DNA blueprint of the virus for the cell to replicate. By introducing new DNA to this virus, it acquires the ability to infect the host with this new DNA and change the host's genetic structure. In 1981, geneticists from Genentech Inc. in California stepped into the agricultural field to isolate the gene in a variation of the foot and mouth virus that produces the VP3, which is code for virus protein 3. This gene causes immunity in test animals without causing infection. Quote, a molecular fragment containing these instructions was then spliced into a plasmid, or small circular collection of DNA, taken from an E. coli bacterium. Then the plasmid and its recombined DNA were inserted back into the E. coli. Not only did the recipient bacteria begin cranking out VP3, but all their offspring produced the protein as well. Quote, as safe as this technique is purported to be, it has significant potential for a serious backlash. Cancer-causing genes were added to a virus which infected several test animals. It was a weak virus, meaning a stronger one could cause much more damage. Gene splicing could lead to radical viruses, such as the cauliflower mosaic virus. Quote, nevertheless, new research continues to raise questions about specific techniques used to create GM crops. One such questionable technique is the use of viral promoters, such as the cauliflower mosaic virus. This is used to stimulate the gene splicing process. According to the OCA, Rowett Institute's Dr. Arpad Putzai discovered a strain of cauliflower mosaic virus stimulated GM potatoes that is poisonous to mammals. The genetically modified potato strain reportedly damaged the stomach linings and internal organs of lab rats. End quote. Genetic modification, especially that of gene splicing, has the potential to benefit the scientific world or to cause great harm to the world as we know it.